Hi, I'm Scott Koenigbauer, and thanks for tuning in to the studio recording of my dissertation defense. So in this video, I'm going to describe 4 research chapters from my dissertation, Dimensions of Reproductive Diversity in Great Lakes Fishes. I have plenty of acknowledgments that need to be covered over the course of my PhD. However, to keep this video reasonably short, I'll just sort of skim through. I'm thankful for my PhD committee, all of who were very helpful and supportive throughout my PhD process. While working in Thomas's lab, I've interacted with a wide variety of student staff. I'm really thankful to have learned from all of them and learn about their interesting research projects. I'm thankful for my family who supported me and been my sort of emotional backbone throughout the way. Throughout this presentation, I've listed all of the research collaborators as well as agencies that have supported each corresponding project. And last, research chapters for my PhD were funded both by the U.S. EPA Great Lakes Restoration Initiative as well as the Purdue Department of Forestry and Natural Resources. So when developing my dissertation, I needed to determine a thread that could summarize sort of the course of all of my research that I worked on, and I landed on reproductive diversity. So my definition of reproductive diversity in a very basic sense is the variation of features relating to reproduction. So again, this is an intentionally broad or very basic definition, and it can be used to describe many different aspects or configurations of research. The 4 dimensions or aspects that I cover in my dissertation chapters include 1. The diversity of reproductive habitat, 2. The diversity of spawning group genetics, 3. The diversity of spawning behavior, and 4. The diversity of size between sexes. So for my first chapter, I characterized the spawning utilization of a restored reef in Saginaw Bay, Lake Huron. This research was funded through the Great Lakes Restoration Initiative and was supported by numerous agency collaborators, including from Michigan Department of Natural Resources, Michigan Eagle, and the U.S. EPA. So when thinking about the diversity of reproductive habitat, one concept that is important is that large populations may benefit from a variety of spawning habitats. One useful framework to think about the benefits of spawning habitat variety is the portfolio effect, whereby recruitment is more robust to interannual variation in conditions when numerous habitats are available. The portfolio effect framework is very similar to how you may manage money or financial investments. So if you were going to invest a large amount of money, it would probably be safer to invest it in numerous stocks, because if one of those stocks failed, you would lose all of your money. We can think about fish spawning habitat similar to stocks in your investment portfolio. So if you have a population that's very dependent on just one spawning habitat, then if something were to go wrong at that habitat in any given year, you would potentially be vulnerable. Your population would be vulnerable to negative changes or loss as well. Tim Sesterhen was a postdoc in Thomas's lab before I started at Purdue, and one of his projects was to model larval fish movement throughout Saginaw Bay. And what he found was that interannual changes in weather patterns, so different weather patterns from year to year, favored recruitment from different spawning habitats. So what this shows is that depending on the conditions of a given year, different spawning habitats in Saginaw Bay may be more important for that year's given recruitment. So again, this is sort of supporting or implying that there's a need for habitat diversity in Saginaw Bay. So the habitat that I focused on in this chapter is rocky reefs. Numerous lithophilic species utilize rocky reefs in the Great Lakes. They're very beneficial for incubating fish eggs because they provide shelter with their interstitial spaces. as well as in those spaces there may be potentially improved oxygenation for those incubating embryos. Historically, rocky reef habitat was very abundant in the Great Lakes. However, through various anthropogenic stressors, rocky reefs have become degraded for fish utility in many areas of the Great Lakes. So here, for example, in Saginaw Bay, you can see two images of unrestored or degraded reefs in Saginaw Bay that have been infilled with sediment as a result of land use change. Between 2014 through 2016, Nick Calais, who is a master's student in Thomas Hook's lab, examined fish utilization at 4 unrestored reef sites throughout inner Saginaw Bay. Ultimately, what he found was that walleye and lake whitefish were still spawning on these degraded habitats despite them being covered in sediment. So it suggests that if you were to restore these habitats, potentially you could benefit the embryos or the offspring of those species, that those species spawn at those sites. Following those pre-restoration surveys, in tandem with sedimentation models as well as stakeholder meetings, managers decided to use Great Lakes Restoration Initiative funding to restore Koenigbauer Reef. This reef restoration is positioned 16 kilometers or about 10 miles north from Bay City offshore, and you can see in the bottom right the footprint of the reef. It's basically a 2-acre or 0.8 hectare rectangle of supplemented rock, with the northeastern half being composed of granite and the southwestern half being composed of limestone. Following the restoration of Koenigbauer, I was charged to conduct a post-restoration utilization survey at the site. So I standardized my methods to the pre-restoration surveys so I could make before and after comparisons. And again, in the fall, I sampled lake whitefish from November through December, and in the spring, I sampled spawning walleye between March and May. The two overarching objectives for these spawn utilization surveys was one, to assess the reproductive usage of the reef by native species, and two, to evaluate the egg incubation environment as well. Just to contextualize our sampling locations, if you look at in the top right panel of this map, you can see the gray rectangle represents the actual restored reef. So we deployed gear on top directly on the restored reef site. However, we also sampled at 2 unrestored control sites, one that was distant about 1 kilometer to the southwest and one that was directly adjacent to the reef. The first component of our first objective was to examine spawner presence. And to do this, we used overnight large mesh gill nets where we would capture spawning lake whitefish and walleye. Overall, what we found was that both species were captured during their respective spawning seasons, and we found that catch per unit effort, which is along the y-axis of these plots, was relatively consistent among the restored and unrestored sites in our post-restoration survey. Further, if you see the black values of mean CPUE here, those are from the pre-restoration surveys, and again we found not really a huge change in the catch rate or relative abundance of spawners before or after restoration. So here what this is showing is that both species were present prior to restoration, and the new reef or the reef restoration didn't necessarily attract new spawners to the area. The second component of this survey was to assess egg deposition rates on the reef. And to do this, we use benthic egg mats. Here in the top right, you can see a picture of egg mats. They're a furnace filter material that's wrapped around a steel frame, and they'll soak along the bottom and passively collect eggs that were depositive above them. And we sampled equally at our restored reef as well as at our unrestored sites with these surveys. Again, similar to spawner presence, we found that lake whitefish and walleye were both depositing eggs directly on the restored reef in all 4 of our survey seasons. One interesting finding was that we actually saw higher egg deposition rates at our control sites than on the restored site, and we think this may reflect gear bias, where eggs that are laid over the restored site may be retained within those interstitial spaces between the rocks. However, eggs that are laid away from the reef may be more likely to drift along the bottom and be collected by our mats. More qualitatively, if you just look at the peak deposition rates observed during the post-restoration survey as well as during the pre-restoration survey, we found a very notable increase in peak egg deposition rate for both species. So the presence of spawners may be consistent pre and post restoration. However, the egg deposition surveys showed that they're actually using the area to lay eggs a lot more readily after restoration. For our second objective, which again was to evaluate the egg incubation environment at the reef, our first component was to survey potential egg predators. And to do this, not only did we capture large potential egg predators in those overnight gill nets, but we also deployed additional daytime micromesh gill nets to sample smaller individuals. And any potential egg predator that was captured in the field was flash frozen, and then stomach content analyses were conducted in the lab. Overall, we captured a variety of potential egg predators with these various gill nets. However, it was round goby, yellow perch, and white perch that predominantly appear to consume eggs at the reef. Most notably, it would be the invasive round goby. They were captured almost exclusively on the restored reef. They really liked those rocky interstitial spaces. And 65% of those round gobies captured did have native fish eggs within their stomach contents, and at a relatively high volume, on average 77 eggs per stomach of round gobies. The second objective of our egg incubation environment survey was to assess water quality. And to do this, we used archival temperature and dissolved oxygen loggers, and we validated the measurements of those loggers using a YSI oxygen probe. If you direct your attention to this left column of figures, you can see dissolved oxygen, and overall what we found was that the concentration or saturation of dissolved oxygen never fell below a threshold that would be potentially negative for incubating embryos. So overall, we concluded from this that the water quality was sufficient for native fish eggs to develop. So some conclusions from Chapter 1. First, we found that native species, so lake whitefish and walleye, both quickly adopted the reef restoration in Saginaw Bay. So these surveys took place just 1 to 3 years after restoration, and we already found both species using it to spawn. Second, we also determined that the reef environment appears to be suitable for egg incubation. However, it is somewhat concerning that round gobies also appear to have adopted the reef and may be actively preying upon native fish eggs in the area. More broadly, this research was part of an adaptive management strategy. So the construction of Coreyon Reef was really a test to verify the effectiveness of the restoration strategy before making a larger investment in the bay. And last, thinking about the diversity of reproductive habitat, we suggest that further reef restoration may bolster the Saginaw Bay portfolio by diversifying reproductive habitat. The second chapter of my dissertation was assessing the spatio-temporal genetic variation in the recovery of Saginaw Bay walleye. This project again benefited from Great Lakes Restoration Initiative funding and was also supported by the Purdue Department of Forestry and Natural Resources. Here we received a lot of samples from various agencies and entities, including Michigan DNR, Ohio DNR, University of Wisconsin-Stevens Point, and Wisconsin DNR. So again, this chapter is considering the diversity of spawning group genetics. High standing genetic diversity may allow for adaptive capacity to ecological change. So if you have greater genetic diversity, your population may be more robust to changes in the ecosystem through time. When a population's abundance crashes, we'll often see bottleneck effects, which may yield low genetic diversity, leaving you with low abundance and low genetic diversity in your population. I suggest that different rehabilitation strategies need to balance the increases in abundance and genetic diversity. So certain management actions may be very effective at increasing population abundance, but may have little effect on population genetic diversity, whereas others may be more effective at improving genetic diversity without necessarily having as large of an effect on population abundance. So the two management strategies that I'll sort of be comparing in this chapter include supplemental stocking and habitat restoration. Supplemental stocking is often very effective at recovering population abundance. It's been widely used in the Great Lakes for decades. However, there is some evidence that when you stock, there may be an initial reduction in genetic diversity because the stocked individuals have a competitive advantage over the native or natural individuals in the system. Further, I suggest that the changes in genetic diversity or recovery of genetic diversity in a bottlenecked population may be relatively slow under supplemental stocking, just given the time scale of evolutionary processes. The second management strategy covered in this chapter is habitat restoration, which we just showed in chapter 1 may aid in the abundance of population recovery. However, there's very little evidence of the effects on habitat restoration on genetic diversity. In fact, the most commonly cited evidence of effects of habitat restoration on genetic diversity involve dam removals, which may be, depending on the system, may be less similar to the portfolio effect that we're really interested here in Saginaw Bay. I suggest that habitat restoration could potentially increase the contribution of population subgroups to overall recruitment. So if you have potentially reef spawning individuals at a very low abundance, if you increase their abundance, they may support greater genetic diversity in the population. Again, I'm thinking about Saginaw Bay walleye with this research. I'll just do a little quick characterization of the history of the population. It was once a very abundant and thriving fishery, and then following World War II, walleye were actually nearly extirpated from Saginaw Bay, and their population abundance was ultimately restored through management activities such as stocking. There was a 30-year stocking program between the 1970s and the early 2000s, and we found that it widely did help the walleye population recover. So here in this figure, you can see between the mid-'80s up until the mid-early 2000s, the walleye population abundance was hovering at about 2 million. And after ecological changes, especially after alewife crashed in the system, we saw just this massive uptick in walleye abundance in the system, where their abundance actually now exceeds some management goals. I'll pause here and just talk about some of the different brood sources used for stocking Saginaw Bay during that 30-year period. They especially started using brood source from Muskegon River, which is a tributary to Lake Michigan. That was between the mid-1970s all the way up to the early 1990s. That was really the predominant brood source that was stocked into the area. However, following that to the mid to late 90s, they just began capturing individuals directly from Saginaw Bay, rearing their eggs in a hatchery and restocking them back out. There's some evidence that the recruitment of Saginaw Bay walleye is almost entirely dependent just on the Tittabawassee River spawning habitat. So thinking about that portfolio effect again, highly dependent on just one spawning investment. And as a result of that, there's been a recent emphasis on reef restoration to try to bolster that portfolio. So the purpose of this project was, again, to assess Saginaw Bay walleye population genetics, and we had two main objectives for the chapter. The first being to assess the contemporary spatial genetic diversity of Saginaw Bay walleye, and the second to be assessing the temporal genetic diversity both during and after that stocking period. To do this, we use a GT-seq panel developed by Peter Euclid that is especially effective at differentiating Great Lakes walleye stocks. So when you use this GT-seq panel, You receive sequence data or genotypes at 500 loci, and for this project we subsequently filtered those sequence data according to their quality. We used various sample sources for our genetic analyses, including we extracted DNA directly from tissue that we sampled during the chorion reef surveys from Chapter 1. We also received archived spine and scale samples from the Michigan DNR that we extracted DNA from. And then finally, because we used this GT-seq panel, we could also incorporate data sets from previous studies in previous parts of the Great Lakes. So the first objective, again, was to cover contemporary spatial genetic diversity. And primarily we were interested in comparing the chorion reef spawning walleye to walleye that spawned in the Tittabawassee River. So sort of a reef versus river spawning or genetic comparison. And we hypothesized that there would be a distinction between reef and river spawning walleye, similar to what has been seen in western Lake Erie. We were also interested in comparing chorion reef walleye to those of Muskegon River, especially those that were captured in 1975, because those reflect the predominant brood source that was used during the early parts of that stocking period. But second, assuming that chorion reef walleye would be genetically distinct from the Tittabawassee River, we also wanted to incorporate alternate population sources for chorion reef genotypes. So we could compare, okay, what are the potential strains or genetic similarities that reef spawning walleye in Saginaw Bay may share with other areas of the Great Lakes. Just quickly to summarize, we had walleye genetic samples from 9 locations in the Great Lakes that amounted to just about 740 observations. So I'm pretty new at doing genetic analyses compared to the more seasoned molecular biologists, but one of my first impressions is that statistical analyses of genetic sequence data reminded me a lot of Taco Bell, where you could use a lot of the same ingredients to develop some very different products. And so here I'll give you a sample of a few of the products that we were able to produce using those 500 loci from the GT-seq panel. First, we made pairwise FST comparisons among locations, and we tested whether they followed the isolation by distance pattern. So here in the figure along the y-axis is FST. A higher FST value suggests that you have greater genetic differentiation. And along the x-axis is the distance between those pairs of locations in kilometers. And these were calculated as a fish would swim, so connected waterways. And here, this positive trend where you have greater genetic differentiation at further differences, again, does match that genetic isolation by distance pattern that we expected. However, the outliers from this analysis were particularly interesting too. So if you look at the largest negative outlier, Muskegon River's pairwise comparison with Tittabawassee River, we found that they were much more genetically similar than would be predicted according to their distance. Next, we use a neighbor joining tree. This sort of groups and pairs your populations that you've sampled, and we use 1,000 permutations and tested the consistency of each node or branch. What we found with the neighbor joining tree is that it produced roughly 3 genetic groups that were fairly consistently separate from one another. The first was that Tittabawassee and Muskegon River walleye seemed to be very similar about 90% of the time. So stocked Muskegon River walleye are very similar to natural river spawning walleye in Saginaw Bay. Second, we found that chorion reef walleye were most similar to walleye captured at other unrestored reefs in Saginaw Bay. And then third, we found that all of our Lake Erie walleye seemed to group together very consistently. Third, we looked at genetic admixture among some of the populations that we sampled. So we used a structure analysis for this. Here in the top figure, each of those very skinny bars represents one walleye individual. And how this works is you assign the genotypes of the loci included in your analysis to 3 parsimonious clusters. So here we can see, depending on your location, how much or what proportion of your genotype is assigned to each of those clusters. To walk you through the structure plot, on the left we have the Sandusky and Maumee rivers, and we see these are dominated almost entirely by clusters 1 and 3, with not very much of cluster 2. So we can assume that 1 and 3 are really reflective of the Lake Erie genotype. Then if you look all the way to the right of that plot, you can see Muskegon River walleye are mostly dominated by cluster 2, or sort of our Muskegon cluster. When you look at the Tittabawassee River, it had a little bit more mixture with some of those 1 and 3 clusters from Lake Erie. However, it was still predominantly mostly assigned to cluster 2, similar to Muskegon River. And then in the middle is Coreyon Reef, and we see almost a consistently even assignment of all 3 clusters. So this is showing us that there was admixture of strains that were using the Coreyon Reef location, some similar to Lake Erie, while others were similar to Tittabawassee and Muskegon River. For our temporal genetic diversity assessment, again, we were interested in the changes in genetic diversity both during and after the stocking period. So again, we were examining trends, and we did this by making comparisons among year classes. So in this table, you can see there are 9 year classes from which we had samples of Saginaw Bay walleye in the Tittabawassee River. And we also had samples from the Muskegon River from 1975 and 2002. So we were interested to see, did Tittabawassee River walleye become more different from Muskegon River years after stocking? And further, were year classes, more recent year classes of Tittabawassee River becoming more distinct from those earlier year classes of Tittabawassee River? Overall, we found just a marginal increase in differentiation between the Tittabawassee and Muskegon walleye. Further, if you look at this plot, each of these points represent pairwise FST comparisons, just like what I showed you for the spatial analysis. And we found just a very marginal increase in FST scores when you used pairs that were a greater number of years diverged from one another. In fact, these vertical bars represent what we would expect for genetic drift under various conditions or various affected population sizes. And because all of our observations or pairs appeared to fall within those confidence intervals, we attributed all of the changes just to genetic drift. We also, again, used the structure plot for our temporal analysis, and we found relatively consistent admixture patterns over the various year classes in the Tittabawassee River. So not really much change at all, and the same could be said for the Muskegon River too. So some conclusions of those genetic analyses. First, we found that Saginaw Bay reef spawning walleye are at least somewhat distinct from those at the Tittabawassee River. They appear to have a lot more admixture with the Lake Erie strains than those from the Tittabawassee River, which are very similar to Muskegon River. Again, Tittabawassee River walleye genotypes still reflect those of the Muskegon River brood source 50 years after the first stocking event. So according to evolutionary time, this is very short and maybe unsurprising, but just thinking more about the lifetime of fish management, stocking, you're not likely to see much change in your genotype of a population through time after stocking. Overall, this chapter shows that restoring reproductive habitat may have more potential to increase genetic diversity than other management strategies such as supplemental stocking. My third chapter was looking into how spawner size influences the reproductive phenology of a freshwater fish. This project was funded by the Purdue Department of Forestry and Natural Resources, and we were supported through various state agencies that provided data, such as Ohio DNR, New York State DEC, Michigan DNR, and Wisconsin DNR. So this chapter, we're thinking about the diversity of spawning behavior, and the behavior that I chose for this chapter was the time that an individual fish spawns, or hereafter I will call that reproductive phenology. Reproductive phenology can greatly affect the experience and potential survival of offspring. So if a parent fish spawns either relatively early or relatively late in a given spawning year, its offspring are likely to hatch at different times and may experience more or less suitable habitats or environments when they hatch. And therefore, the time that a mother spawns, for example, may greatly influence the survival of her offspring. We also know that reproductive phenology can vary both at population and individual levels. So thinking about population level variation in reproductive phenology, these are mostly related to spawning cues, such as temperature and photoperiod. However, at an individual level, there's also evidence that reproductive phenology can vary with maternal effects, where larger or older females may spawn at a different time than younger or smaller conspecifics. The evidence of individual variation in reproductive phenology is limited, especially in males. And a lot of this evidence are really just anecdotal accounts suggesting that the largest individuals of a population spawn the earliest. The reason why there is such limited evidence of this individual level variation in reproductive phenology is that studying fish phenology is uniquely challenging. Unlike terrestrial organisms, we often can't visually monitor fish reproductive behavior. And instead, we often rely on indirect methods to observe them, unless you are researching nest-guarding fishes, which in some cases you can dive and visually observe their behavior or activity above the nests. Some other examples of indirect methods that fisheries researchers can use to study behavior include monitoring migration with fish counters or pit tags, or using acoustic telemetry to track fish movement. While these types of methods are very useful for whatever questions they've been applied for, these methods can also be very limited for other particular questions relating to individual reproductive phenology. So based on logistic constraints, these typical methods are often limited in spatial or temporal distribution, and they also often have smaller sample sizes. Further, methods such as acoustic telemetry often have little information about individual fish, such as length, age, or sex, at the exact time of spawning. Therefore, our goal was to determine whether larger fish of either sex generally spawn earlier than smaller conspecifics. Or in other words, is there individual variation in reproductive phenology according to fish size? And to do this, of course, I studied walleye. And these were a very useful candidate species for this type of analysis because they have distinct spawning events and locations. They spawn en masse or in large aggregations. And it's assumed they have very little mate selection, unlike a lot of past evidence from other taxa in the literature. And then last, and probably most importantly, walleye are a common target for recreational fishers and are intensively managed as a response. And therefore, those management activities yield large data sets that can be analyzed. The two objectives of this study were, one, to assess whether larger fish were captured earlier in annual fisheries independent surveys, and two, whether anglers captured larger spawning fish in a given year. So to do this, our goal again was to be very general, and so we were hoping to find and be able to test observations at a broad spatial and temporal scale. So here you can see a map to the right of all the various locations from which we collected data from numerous agencies, and those observations spanned a pretty wide temporal distribution or range, ranging from the 1940s all the way up to the late 2000 teens. At these locations, agencies applied various fisheries independent collection methods, such as electrofishing, gill netting, seining, or using trap nets. Unfortunately, using this very broad type of data set, we could not directly combine or compare observations across locations or years due to discrepancies, again, within and among those years and locations. So some examples of discrepancies that would confound our analyses included interannual environmental variation or across locations. So for example, reproductive phenology spawning cues may vary over years or over locations. And second, all of these different sampling gears that are used by agencies at various locations have their own biases towards catching larger or smaller individuals. Instead, to incorporate this very broad data set, we instead used effect size analyses, which are very similar to meta-analyses. Ultimately, what we did was calculate correlation coefficients from every year that was sampled at every location, and we transformed them to effect sizes. So here in the bottom right, you can see these are relationships between total length and the day of year that walleye were captured in the Tittabawassee River. We can extract correlations, transform those to Fisher Z effect sizes, and once we've calculated Fisher Z effect sizes from every year within that location, we can then calculate a mean effect size with 95% confidence intervals. So for our first objective, to assess whether larger fish are captured earlier in fisheries independent spawning surveys, again, we had a really broad data set with observations from 13 locations throughout the Great Lakes region, and we also had a very large number of just individual observations as well. Roughly 70,000 female walleye and over 150,000 male walleye observations in our data set. The figures above, you can see each line represents an individual year at a location's relationship between walleye total length and the day of year from which it was captured. And then if you follow those below, remember we extracted those correlations, transformed them to Fisher Z's, and calculated means for each location. And ultimately what we found was that 9 of 13 populations had negative effects of size on date of capture for females and 7 in males. So these negative effects suggest that suggested that larger individuals were captured earlier in their respective spawning years. We also used a random effects meta-analysis to estimate overall mean effects from these populations, and we found again these significant negative effects. For our second assessment objective, we were interested in whether larger spawning walleye were captured earlier by anglers. And to do this, we used creel survey observations from 2 tributaries of Lake Erie from the Ohio Department of Natural Resources. This was a somewhat smaller data set, but nonetheless, we found roughly the same finding. Yes, for both sexes, larger walleye were captured earlier during their respective spawning periods on spawning grounds than smaller walleye. And that was consistent not only just for our location analyses, but also in our random effects meta-analyses as well. So some conclusions from Chapter 3. First, we found evidence that generally larger walleye of both sexes will spawn the earliest. Those patterns were observed not only in fisheries independent survey data, but also in angler surveys as well. These findings have implications both for fisheries management as well as broader general fish ecology. So thinking about fisheries management, this shows that size selective harvest could have effect on the duration of a population's spawning period. So for example, if you use size selective harvest and you capture all of the largest individuals in your population, you may also be capturing all the individuals that spawn the earliest in the population. So you may shorten the duration or window of time that they spawn in. Second, thinking more about general fish ecology, walleye are considered to be, again, a mass spawning or aggregate spawning species where it's not assumed to have much mate selection. However, If you find that the largest individuals of both sexes spawn the earliest, it may suggest that there is some mating structure in these populations where the largest individuals are more likely to spawn with one another. To conclude, a broad size structure of a fish population likely increases the diversity of reproductive phenology. Okay, and finally, my fourth chapter of my dissertation was assessing the conditions for emergence of walleye sexual size dimorphism in an ecogenetic model. Again, funding for this was provided by Purdue Department of Forestry and Natural Resources, and we also thank the Purdue Rosen Center because we used their Bell cluster for these simulations. Okay, so this last chapter was focused on the diversity of size between sexes. And one example of diversity between sexes would be sexual dimorphism. And this is observed all throughout the animal kingdom. So sexual dimorphism is just broadly suggesting that males and females of a given species have some sort of consistent variation from one another. So some examples of sexual dimorphism include body shape. So male and female anglerfishes, for example, have very different body shape. Secondary sex characteristics like bucks and does, white-tailed deer. Bucks obviously have very large antlers. And last, you think about the dimorphism of coloration, especially in songbirds, where often males have maybe more pronounced or vibrant coloration when compared to females. The most common form of sexual dimorphism is sexual size dimorphism. And when I talk about sexual size dimorphism, I may use terms like female biased. And when you say female biased, it suggests that females on average are larger than males. We know the direction and extent of sexual size dimorphism can vary, so it can vary among species. These are results from a paper by Horn et al. From 2020 that I think are really cool, where you can see the direction and strength of sexual size dimorphism varies taxonomically, it varies by environment, it varies by reproduction mode, etc. We also know that sexual size dimorphism may vary among populations. So here we see an example of Eurasian perch, and the extent of their sexual size dimorphism changes according to the latitude at which the population is found. Despite contemporary evidence of sexual size dimorphism or observations of consistent patterns of sexual size dimorphism in the wild, it's difficult to observe the evolutionary drivers that may have led to this pattern. Some hypothesized mechanisms for why SSD is prevalent now include mate selection. So here you see, I think, a long-eared sunfish guarding his nest. Being a larger male sunfish may provide some advantages, making you more desirable for reproduction. Second, it's hypothesized that differential gonadal investment may drive sexual size dimorphism, where females have much greater investment into their gonads than males. Third, it's possible that differential natural mortality could lead to sexual size dimorphism. So variation in metabolic rate or respiration rate relating to general activity levels of the sexes could be an example of different natural mortality between the sexes. And finally, maternal effects or the general patterns that larger, older females have more viable offspring could also be a driver for sexual size dimorphism as well. These hypotheses imply not only the direction of sexual size dimorphism, but it's also unknown whether they act in isolation or synergize or antagonize with one another. When thinking about sexual size dimorphism, it's important to note that the growth of an individual is often affected by when they sexually mature. So the trajectory of their growth may vary according to the time in life when they mature. And this is because once an individual is mature, they're suddenly dividing their energy between growth of somatic and gonadal tissues. Therefore, studying sexual size dimorphism often benefits from considering age at maturity because it again can affect the overall trajectory of when individuals or how large individuals grow. In fisheries, one way we describe the variation in when fish mature is thinking about maturation reaction norms. So here, this diagram on the right, in blue, you can see the growth trajectories of an individual under good or poor conditions. So the maximum size that they reach may be related to the environment that they live in. And in red lines, these are heritable maturation reaction norms, whereby if an individual grows to that certain size at that given age, then they're likely to mature. So here you can see not only can your growth trajectory depend on your environment, but also the time at which you become sexually mature can vary according to your heredity. So for this chapter, I used an ecogenetic model that simulates cool water fish evolution. It was developed by Wei-Yu Wang, who is a PhD student for Thomas Hook. And it's effective at simulating evolution in very large cool water fish populations. Because it's an ecogenetic model, it incorporates not only population dynamics, but also heritable or genetic traits. In this model, the genes that are passed from parents to offspring are inherited growth parameters, and it uses a codominant inheritance strategy or mode where the parameters of mothers and fathers are averaged in their offspring. What I did was I reparameterized this ecogenetic model to simulate the evolution of a walleye population for about 2,000 years. And while I ran these simulations, I tested various evolutionary hypotheses for sexual size dimorphism. So I ran a baseline simulation, and then I ran simulations where males and females had different gonadal investment, different natural mortality. I applied maternal effects to females, and then last I ran a combined model to see if those hypotheses or those mechanisms would synergize with one another. When I received outputs from those models, I looked at the maturation reaction norms of the population after 2,000 years for both males and females. And I also compared growth parameters between those sexes as well to determine SSD. Okay, so first we found from our baseline simulations parameters were exactly the same between males and females, so not surprisingly after 2,000 years there was no pronounced sexual size dimorphism. When I coded different gonadal investment where females spent 4 times as much energy on gonadal investment as males, we actually didn't see sexual size dimorphism emerge, but we did see that females needed to grow to a larger size at younger ages to mature. So it suggested sort of more growth in early life and later maturation for females. When I increased male mortality to different instantaneous mortality values, we found the first evidence of sexual size dimorphism. Whereas male mortality increased, they grew on average more and more smaller than females. When I included maternal effects in the model, again, this also supported sexual size dimorphism, again, which was female biased. Females grew larger than males on average. And then finally, when I combined the hypothesized mechanisms, we found that those mechanisms did in fact synergize and lead to our strongest extent of sexual size dimorphism, again where females were larger than males. So based on simulations of walleye evolution over 2,000 years, we did find emergence of SSD under various hypotheses. However, the maximum sexual size dimorphism that we observed was only a 5% difference in size between males and females. Versus in the wild, walleye SSD can range from 10 to 25% difference in maximum size between sexes. The reason why the percent SSD was smaller in our model is likely because we were isolating the genetic component, where we weren't incorporating ecological parameters or experiences or environments that may further drive differences between sexes. So, for example, there have been observations that walleye of different sexes may occupy different habitats or temperatures, which could affect their their growth and metabolism. And next, there's also differences in the consumption rates of the sexes. So females have, in this example, much higher consumption rates than males. So again, that could further drive the difference of sexual size dimorphism as well. So we looked at those natural evolutionary conditions. However, what would fishing do to affect the extent of sexual size dimorphism? So we know that fisheries-induced evolution can affect population growth, maturation, and reproductive investment. And oftentimes, heavily fished populations demonstrate earlier reproduction and reduced overall growth. So for example, here this figure shows a yellow perch fishery where the gray background represents when commercial fishing occurred. And what it shows is that the age or length at maturity of yellow perch in this population shot back up as soon as commercial fishing was closed, suggesting that commercial fishing was causing earlier maturation and shorter overall growth. So thinking about fisheries-induced evolution, we ran those same simulations over again. However, we also, after 2,000 years of natural evolution, also applied 1,000 years of fishing under different fishing regimes. What we found was that when we used unregulated fishing, where any recruited individual had fishing mortality, we found that the extent of sexual size dimorphism was reduced, where males and females became more similar in size. And this was mostly driven by the largest females are being captured from the population. When we applied slot size fishing, we saw the greatest reduction in sexual size dimorphism. But under this condition, it was actually that males appeared to grow larger because once they surpassed a certain size, they were no longer exposed to fishing mortality, so the largest individuals of both sexes were protected. And last, thinking about size-selective fishing, we found that sexual size dimorphism was relatively consistent, both before and after that fishing period. So some conclusions from Chapter 4. Using an ecogenetic model, we found support for 2 of 3 evolutionary hypotheses for female bias, sexual size dimorphism, and walleye. And those were, again, differential natural mortality, where males had higher natural mortality than females, and also maternal effects. We also found that sexual size dimorphism was maintained under fishing, but there were some changes in the extent of that sexual size dimorphism once fishing was applied, and further we saw some evidence of changes in the population size structure as well. So for example, we found that the various fishing regimes may remove the largest or oldest individuals from a population. And finally, we conclude that sex-specific variation in reproductive biology contributes to the diversity of sizes in that fish population. So some conclusions from my broad dissertation. Again, this research centered around how reproductive diversity has many dimensions, but I suggest that all dimensions or aspects of reproductive diversity are valuable when you're considering the stability of a fish population. In Chapter 1, I demonstrated how reef restoration or habitat restoration may be a viable management tool to benefit native fish spawning. In my second chapter, we found that habitat restoration may have greater potential to benefit population genetic diversity than other recovery strategies such as stocking. Third, I showed in my third chapter that population size structure affects reproductive phenology and protecting the structure of protecting a population's size structure may broaden the spawning duration on a year-to-year basis in that species. And last, through ecogenetic models, we ran simulations of walleye evolution that supported hypotheses for evolutionary mechanisms of sexual size dimorphism. And that sexual size dimorphism then contributes to individual size variation. So here are the references that I covered throughout my presentation. And with that, thanks for listening.